Hi everyone, thanks for tuning in. Today I'll be talking about how fossil biomolecules reveal the physiology and paleobiology of extinct amniotes. Metabolism drives organismal function, and this has always life itself. Throughout the history of life, there have been numerous innovations of metabolic pathways. However, amniotes have taken their metabolic capabilities to new extremes. We know that mammals and birds independently evolved exceptional metabolic rates based on different molecular upregulation strategies. The metabolic rate is traditionally defined as the volume of oxygen respired relative to the animal body mass over a constrained interval of time. While we know of facultatively elevated metabolic rates in certain lizards and snakes, the metabolic rates of mammals generally exceed those of the pterosaurs and are only surpassed by the avian metabolism. It is not a coincidence that we find the highest metabolic rates in the animal kingdom associated with endothermic warm-blooded taxa. A cascade of biochemical cycles, including glycolysis, the Krebs cycle, and the electron transport chain, facilitate the conversion of respired oxygen into chemical energy, and are coupled to a process called oxidative phosphorylation that releases some of this chemical energy in form of heat. The degree of coupling between these processes is different in warm and cold-blooded animals and defines the fundamental differences between mammalian and avian endothermy. The generated metabolic heat is the key to active thermoregulation, which represents a physiological advantage characterizing all endotherms. For this reason, the metabolic performance and resulting thermal strategy represent a powerful lifestyle proxy and have previously been suggested as a key advantage, explaining not only the survival of mammals and birds across the KPG mass extinction event, but also their subsequent rapid diversification. Unfortunately, this theory is really difficult to test, as we do not know much about the metabolic capabilities of extinct amniotes. Body temperature estimates based on isotope fractionation and histological assessments of growth rates represent downstream effects of metabolic capabilities and therefore do not allow direct inferences of metabolic and thermal strategies. Mammalian-style endothermy has been proposed to have evolved in their early synapsid ancestors, and avian-style endothermy has been suggested to have emerged in theropods, in dinosaurs, or even in archosaurs. To resolve this uncertainty about the onsets of metabolic endothermy, we need a direct proxy. I hypothesize that metabolism leaves molecular signatures that have the potential to fossilize and can reveal the evolution of physiology and paleobiology in extinct amniotes. To find a suitable metabolic marker, we have to take one step back to understand which biomolecules actually leave informative traces in the amniote fossil record. Previous research has taught us that insoluble kerogen can survive in deepest time. Back in 2018, my team and I conducted the first comprehensive screening of vertebrate fossil organic matter to reveal that endogenous proteins, lipids, and sugars preserve as fossil organic matter through oxidative cross-linking reactions mediated through so-called reactive carbonyl species. In 2020, my team revealed that fossil biomolecules retain biological signatures despite chemical alteration, informing about biomineralization, tissue types, phylogeny, and metabolism. What we did not answer was how this metabolic signal preserves in fossil bone. So let's make the next step today. Here's the theoretical approach. On a microscopic level, bone has a proteinaceous skeleton, the extracellular matrix, that holds mineral crystallites and bone cells in place. Every amniote cell contains mitochondria, tiny organelles that are often referred to as the power plants of the cell. The key biochemical cycles involved in molecular respiration act in the inner mitochondrial membrane, right here. Corresponding to the amount of oxygen that's being converted, electron transfer chain and oxidative phosphorylation generate as accidental byproducts reactive carbonyl species. These reactive oxygen species permeate membranes by forming reactive carbonyl species, which in turn trigger metabolic cross-linking, both within and outside the cellular realm. The total accumulation of resulting oxidative protein damage in the extracellular matrix corresponds ultimately to the volume of respired oxygen the metabolic rate, and is increased by adding a surplus of diagenetic crosslinks during biomolecule fossilization. My approach proposes that the relative quantity of crosslinks, once calibrated with extant taxa, allows to retrocalculate the metabolic rate of extinct amniotes and their resulting thermal strategies. I've selected a taxonomically diverse set of modern and fossil amniote longbones. The key requirement is preservation of endogenous organic matter, and to ensure that, I chose fossils based on the criteria I've identified in 2018. Brown to black stained fossils preserved in a light-colored sediment matrix suggest molecular preservation. Both modern and fossil samples were subjected to the exact same analytical routine, including surface cleaning with ethanol and non-destructive in situ Raman microspectroscopy. Raman spectroscopy relies on a laser beam that excites sample molecules and triggers chemical bonds to vibrate in response to their molecular environment. These vibrations are characteristic for functional groups, small molecular units, and lead to Raman-style scattering of the incoming laser light. This signal is amplified and ultimately detected in form of spectra, in which every peak represents a characteristic functional group. I'm showing you here a plot of all fossil bone spectra in this dataset, as they contain, thanks to diagenesis, less molecular information than their modern analogues. As you can see here, there are two areas of major variability in these spectra, reflecting characteristic crosslinks. Once we extract relative intensity values for all modern and fossil bone spectra at the positions 1, 2, and 3, we can form a ratio by summing up the signal intensity at band positions 1 and 2, which reflect sulfur and nitrogen crosslinks, divided by the intensity in band position 3, which represents intact peptide bonds, and thereby corrects for diagenesis. Before we can go on and test to which extent these ratios may reflect metabolic rates, we have to make sure that the organic signals in the fossil samples are not the result of contamination. To do so, I ran whole spectral data in a chemospace principal coordinate analysis to see to which extent sample and sediment clusters overlap in their composition. Fossil taxa are significantly distinct from sediments in their organic composition, and further assessments testing for contamination with glues, microbes, and modern fauna, here shown at the bottom, reveal an endogenous nature of the organic signatures. That's really exciting. So now we can critically interrogate our cross-link to protein ratios. I'm sure you remember the theoretical approach to reconstructing metabolic rates, here again shown on the top right. If the obtained ratios contain indeed a metabolic signal, I would expect to find a sorting of taxa based on their approximated metabolic rates when plotting them against published body mass estimates. I would also expect to see that fossils have substantially higher ratio values due to their diagenetic surplus and crosslinks. Fossil data, here on the top right, and modern data on the bottom right meet both expectations. Yay! I used the pseudo-fossil mammals, birds, and lepidosaurs with close extant relatives to scale spectral cross-link to protein ratios to published metabolic rates. And I separately scaled the ratios for all extant amniotes to reflect metabolic rates as well. Here, the y-axis represents log-transformed metabolic rates, while the x-axis reflects log-transformed published body mass estimates. These are the modern amniote data points. Generally, Raman metabolic rates tend to minimally underestimate the actual metabolic rate. Here are the fossil data points. Based on the complete amniote dataset, I wanted to assess the resulting thermal strategies based on metabolic thresholds. To do so, I calculated a 95% confidence interval based on all modern and fossil crown mammals and birds, shown here in red. Further analyses reveal a clear separation between endo and ectotherms based on their metabolic rates. Next, I've plotted the calculated metabolic rates and thermal strategies on an amniote consensus phylogeny and reconstructed the ancestral states. Let's look at synapses first. High metabolic rates and resulting endothermy evolved in the mammalian stem, but were not present in the analyzed pelycosaur with organic preservation. As here reflected in the quite similar color shades of mammals, extinct and extant ones sampled across the phylogeny, mammalian endothermy has always been very constrained in the spread of metabolic rates that are realized contrary to endothermy based on the avian mechanism. In diapsid amniotes, elevated metabolic rates consistent with endothermy evolved independently in plesiosaurs, varanid lizards, here reflecting facultative endothermy, and in ornithodurans. Drastic reductions of the ancestrally high ornithoduran metabolic rate are observed in all major clades of ornithischians, where they result in secondary ectothermy, and these reductions cannot be explained through allometric effects. Based on this data set, I recovered avian-style endothermy as a trait ancestral to all ornithodurans. Should be noted that the avian metabolism preceded thawed flight, manrapture and egg brooding, theropod carnivory, and appears to coincide with the evolution of filamentous and tegumentary features if they are actually homologous. Ectothermy in many ornithischians has fundamental implications for their paleobiology, requiring behavioral thermoregulation in modern taxa that's often realized through sunbasking or seasonal migration. Ornithoduran endothermy reflects a uniquely wide range of metabolic rates realized through the avian-style metabolic biochemistry when compared to mammalian endotherms. Metabolic upregulation and endothermy in non-ornithoduran diapsids, like for example the plesiosaurs, falls outside the range of avian-style metabolic regulation and may represent a fundamentally different approach to thermoregulation. Looking at the bigger picture, I found a significant correlation between the metabolic rate and the energetic cost of locomotion in extinct and extant amniotes. Even though it has been suggested that elevated metabolic rates provide an advantage in the survival of major environmental perturbations, such as the terminal Cretaceous mass extinction event, we find a number of taxa with extraordinary metabolic capabilities that went extinct during the Capra mass extinction. I've labeled here the clades that survived into the recent. That suggests that factors other than metabolism determine the fate of amniotes during major mass extinction events. In summary, I found that endogenous fossil organic matter preserves biological information such as metabolic signals. By analyzing extinct amniotes, we found that metabolic rates and resulting thermal strategies were highly variable traits. Endothermy evolved at least 3 times independently in mammals, non-ornithoduran diapsids, and ornithodurans. Spectral data can be easily converted into metabolic rates, and this new proxy allows for the direct integration of metabolic signals in modern and fossil amniotes. Beyond the applications to fossils, this new metabolic marker offers a non-invasive method to fill current gaps of knowledge on the metabolic capabilities of protected extant taxa based on skeletal materials in museum collections. By working collaboratively towards extracting more information on how life reacted to past ecological and environmental challenges, we can make informed decisions to protect biodiversity in times of global climate change. I'd like to thank you very much for your attention, and I'm happy to answer any questions.